Yeah. So, this is one way through which you calibrate cantilevers. What is the stiffness of a cantilever? One way to calibrate them is to just let them vibrate thermally, and those oscillation amplitudes are through equipartition theorem. We say this is a spring with a massless spring with a point mass, and then you equate okay. So, this is a good question, and I hope I get time to do this at tutorials. We can do this in tutorials maybe. Half k c mean square displacement square and then take mean equals to kbt, okay, or half kbt. So, you can drop the half. So, typically if I have a cantilever and the temperature is high, it will do a lot of thermal fluctuations, right, and this is what that equation tells you. The I am having it. Okay, so this is the Boltzmann constant. Okay, so the temperature is driving this movement of the cantilever itself, the whole thing. And if I have a stiff cantilever, this will be less, because the product of this has to be the mean square displacement of the cantilever. If you do grab a time series and then look at the mean square displacement, it will be proportional to kBT. T is temperature, room temperature. So, if I have a stiff cantilever, this is less. If you have weak cantilever, this will be high. So, in a way, this also is a way, looking at this is a way to calibrate the cantilever. And there is a paper which has done this quite elaborately. Too much maths, I am not going there. So, so cantilever calibration also we call thermal tuning, thermal calibration of a cantilever. And we will discuss this more in today's tutorial session. Okay. But this is what it is. That is the most significant thermal motion. Now, if you say if I have a cell, and but cells are actually always not only room temperature, at higher temperatures in our body. So, it's okay for cells and proteins and things, but large fluctuation comes out of this cantilever's movement. Yeah. Right. So, those voltage signals are grabbed by the digital to analog, analog to digital. So, these analog signals are converted to digital, and digital is then plotted with. Yeah, the voltage is basically corresponding to some physical quantity, like the voltage will be either to bending or xy position, right. Those are taken and given to the computer, and these are analog signals converted to digital and then finally displayed as a digital, like a photograph or something. X, Y and Z, all three. Okay, let's start. I think next topic is, but before I go into dynamic AFM, and I do not have a presentation to show, but we can discuss this in a informal way. Question is, how is the AFM image taken? And the two questions, one, AFM image and taken for spectroscopy, or in other words, how force is measured as versus distance. So, two things one can do. Suppose this is a sample, that is a compliant soft thing, or maybe there is a cell sitting on, instead of this substrate I have like a cell, a human cell or some mouse embryonic cell or whatever cell. So, and you press on this. Okay. So, now if you press on this, then it deforms, it gets squeezed. How much force we apply to get it squeezed by a few nanometers or tens of nanometers. All of this is done through something called force spectroscopy. Okay. And in here, this is the image, which is the image. Here we grab something called force distance curve. Okay. So, we will, and this force distance curve then can be used to do force spectroscopy and various things, which we will over time, over next 4 days, we will discuss this with great, in great details. So, let us first worry about image and then how force spectroscopy is done. So, we are now cantilever is at angle, laser comes, goes there, everything is made 0. Now, you bring the sample close to the cantilever, and the cantilever initially there is a force of attraction. So, cantilever bends down, but then at one point the cantilever touches the surface, okay, and it touches and you can keep pushing and it can go back. Okay. So, if I really plot the signal in the photodiode, okay, which I will call as the cantilever signal, okay, and if you plot that versus Z, which is essentially the voltage I apply to this scanner, which will keep it expanding and keep it coming up, and that is what is plotted on this graph, this axis. This cantilever signal will correspond to force through a calibration method, right. If you know the cantilever stiffness, you know how much bending of the cantilever corresponds to how much volt, that gives you bending. That bending multiplied by this cantilever's constant gives you the force acting on it. So, essentially on this axis you will have force, and what is this force? This is a force sensed by the cantilever. Okay. Now, suppose this is kind of 0 0 here, okay, this is contact point, and now essentially what happens is, when the force is 0, cantilever is far off, there is no force active, no bending. So, this thing is 0. So, you approach, let us say, come from this side, okay, you are coming in slowly, and what happens now, cantilever bends down, okay. This is how typical force curve look. It will be like that. As you bring the surface, it will bend down. That is the negative force, because attractive force, right, coming down, and it will then from that point, from somewhere where the slope of this force versus distance z, if I look at the slope of this point, when that exceeds cantilever's force constant, okay, k is becomes smaller or equal to the df by dz, which is slope at this point. At that point, so it's going like that, you come slowly, and it is bending, and the rate at which you are doing this and going down, when it exceeds the stiffness of the lever has a jump in instability. So, this jumps to surface vertically. So, it goes down, bends, and just jumps into surface, and then once it is in surface, then you keep pushing backwards, and this will keep going up, okay, crossing 0 of course here, and this is corrected one, and this is what typical force curve look like, and this is where it is it seems like this is a negative z. Now, negative z does not mean anything, right, but what it means is that you are going further and the thing is going up. Once you correct the deflection and everything, it will look differently, but in a typical contact mode imaging, of using AFM, you got, what you do is you basically approach, and if the cantilever is stiff, does not bend too much, stiff cantilever, weak cantilevers bend little bit, and you hit it, and you are loading the cantilever in a backward direction. Okay. So, you are sitting the way, if I can draw this again, this becomes, okay, comes there, at one point it jumps and goes up again, and this will be, the 0 0 is separate now, this will be 0 0, okay, this point, this is where the force is 0 and also the deflection is 0, okay, and whatever that is. Now, if you really convert and everything, what happens is that, this is, suppose I stop at this point or stop at this point, what is the difference? I have loaded the cantilever large here compared to there. So now you can imagine something like your old gramophones. You had this thing going down there and it was sticking there, and when the whole thing rotated, it went up and down, right? And that produced a signal, that is your voice. That's the voice that you hear from gramophone. Very similar thing, or exactly similar thing is done now. You take the thing down and let it touch, push it backwards a little, and now you scan the thing beneath. Okay, and if that has a roughness, this will move back and forth, because the force will be more and less depending on the load, provided the entire substrate, the whole of substrate has the same Young's modulus, the same stiffness. It cannot be that you have a cell in between and a mica or some other substrate. The cell is soft, the other thing is not so soft, and you still scan like this. This thing doesn't hold, right? Because it will then the cell will be squeezed, and when it's not off the cell, it will not be. The loads are different in all of this. So you have to decide at what load, at what force you are going to load the cantilever with, how much you are going to push the cantilever up. This is the contact mode imaging we are talking about. Now this is done. Now the problem is, suppose I load the cantilever and it is about 1 Newton per meter, and you are scanning the surface, and suppose there is a big height. For instance, I am scanning something at this height, and you are touching the surface is loaded back, and you come here, you got this huge guy sitting here. You either topple it depending on how strong it is, or suppose this is like a book, is too tall, but suppose this book is not getting toppled over. So you have to come this and come up again. So load becomes suddenly high. So low load, high load, again low load, and then again high load, and so the whole image is not taken at a constant force or constant loading, and this creates problem, particularly for specimens which have, like this book will be softer compared to the glass, and the load is difficult to then work out how much that whole load is. So what we would like to do is image this whole thing under constant load, so that if I come here, the load is, the cantilever is bent exactly by the same amount when I am here or on top of anything else. And this is done by something called feedback loop. And when we do imaging, which is called a constant load or a constant force image. So how is that done? We have to wait. So along this curve, you either sit here or here, and throughout the image, I don't move from there, which is not possible without feedback loop, because the G is going to change because due to topography. And if you suppose are sitting low on the glass and sitting on a book, the load is going to be different, because the distance is cantilever is pushed back by different amount. So I would like to sit at either my, I decide this is the load, because my sample is so soft, I am imaging sail or something, it's so soft that it cannot bear more than 500 piconewtons of load. So I say, okay, throughout the image do not exceed 500 piconewtons, or throughout the image hold it always at 500 piconewtons. Okay, and this is done by something called a PID loop. So PID loop is, in short, its name comes from proportional integration and derivative loop, derivative controller. But the idea is this: suppose I want to hold temperature of this room constant, what is done? Like we are sitting in AC now, and the temperature outside is different. So we are controlling the temperature here. What do? We put a thermostat. We say the temperature of this room is 25 degrees Celsius, and the thermostat reads the temperature of the room and says, well, 25 is what we want. So 25 is where the thermostat is set, like geyser also. And the temperature, when it is sort of going above 25, it will start the cooling. Okay, it will cool to a point where it will drop below 25 now. Now it just has stopped cooling, but that doesn't happen exactly at 25. There is a room, right? You say, okay, 25 is what you set, but it will go to 30, then you stop, then you start cooling, then it cools down, 25 crosses below 25, comes to maybe 20, and then you stop cooling. So as a result, what will happen? I want to hold the temperature fixed at 25 degrees, but it will keep oscillating between 30 and 20, or maybe you can keep that room little smaller, but it will always keep oscillating like that. So, we say that the temperature of this room is held constant, but it is not really held constant. It is fluctuating, it is going up and down throughout as the thermostat is tuned. Here what we want to do is hold the force constant in a similar way, okay. But the thermostat thing is not active. This is a controller, this is trying to control room's temperature, but this is not active controller. What is the other idea of controlling things at a fixed value? And this is done in physics, it is a controller thing basically. This was first discovered, or rather done by Maxwell, and he used to call it governors, not controllers. Okay, this is doing this for ships. So, the idea is following. Suppose I want to hold a physical like temperature constant, force constant, current constant, okay. All of these quantities you want to hold constant, okay. Then you put it and you hold constant in an active way. What does that mean? That if the temperature or force or the current is moving little bit from that fixed value, we do something to bring it back to that fixed value, and so this is called active. Whereas thermostat in a geyser is a passive controller. It does not do anything, it just measures temperature, and as it goes above certain threshold, it will stop cooling or start cooling, okay. And so this creates a oscillating temperature in a room. We do not want that, we want it all the time at the same fixed value, and this is done through a PID controller. So, how is that done? So, let us say, and this is your, do not have to go into the details, but this is roughly how it is done. So, you will take in cantilever signal, which is force, which is what we want to hold constant. Now, that force is a voltage signal in the end, and PID will understand what voltage is, and so we will call something as a reference. This is the value we want the force to be at. So, I will put this to 1 volt, let us say 1 volt, always 1 volt, and when this force 1 volt corresponds to some force, right, in this whole business, when that is at 1 volt, and in this reference is 1 volt, this also is 1 volt, PID does not do anything, and this PID, the output of PID is connected to AFM, AFM meaning the detection thing that we talked about. So, we can push the G of the piezo up or down or whatever, that is done by the AFM. So, for instance, suppose the force is, I want it to be 1 volt, which is say 1 nanonewton, okay, but the force exceeded and became 1.2 nanometer, and let us say the proportionality is 1, that 1.2 nanometer corresponds to 1.2 volts. So, 0.2 volts extra force, and I want to bring it back to 1 volt. So, what is done? This signal from the PID, which is the difference of 0.2 volt, is taken into consideration by the PID, appropriate signal is generated, goes to the AFM, particularly to the Z piezo, the scanner piezo that we had below, no. What's happening? Why? This thing. So, take time to load, but you saw that the sample was kept on this scanner piezo, right. So, you got the cantilever that is like coming here, once again I had this bent upward at 1 nanonewton, and if I go there it becomes 1.2, the 0.2 difference, which means I have to pull this whole thing down by how much? No, how much you pull down by? Thickness of this book, right, because assuming that the softness both have the same amount of stiffness. So, here if it is 1, there becomes 1.2, I pull this down by thickness of this book, it becomes again 1, and that is what the AFM does, that is what is that is what the AFM does, and it sort of creates this F once again to 1, and then reference is happy, okay, as long as you are on the book, reference is happy till it is on a book. From here 1 volt, 1.2 again brought down to 1, only at the edge little bit of a problem, of course this will take time, right, but right on the book it is again okay. You come off the book, it is again, now you have to push it up by thickness of the book, so that you get back to. So, what happens, you have now scan xy. So, you know the position xy record. At each xy, you know how much you pulled down and pushed up by, and that is your topography. Okay? And that is how AFM image is taken. And now, the beauty of this is that it is at fixed load. So, suppose this book is different material, it's softer, but I still do not want to exceed 1 nanonewton. It will not be pushed up by this thickness, but little less or more depending on if it is softer or harder. You got the point? Like suppose this is a cell, yeah, and I came with 1 volt, and this is now cell thickness is whatever, and the load on it is larger than 1. So, now we said that it has to be pushed back by same amount, that is thickness of a cell, but cells loading behavior is different. For the same amount of deformation, cell will not be same amount of force, cell will not be deformed by the same thing as the glass. So, you pull down by different amount. Okay. So, it will not only give topography, it also can give the mechanical properties of the material beneath it. Okay. But that is little more complex. At the moment, let us understand, if there is a same Young's modulus all over the surface, you will have to push and pull the sample up and down exactly by the amount of the topography over it, and it will produce an image. So, this is how imaging is done under AFM. Any doubts? Yeah.
So, the force that we are saying, this
Yeah.
is the force with which the cantilever is approaching.
Force at which the cantilever is loaded back, and you hold that constant and you scan. So, now if you are scanning, and if the force here is there and here it lows, you basically come down, which is what I gave you the example of the book. So, reference force is the your choice. You say 1 Newton is good for your sample, or 100 piconewton is good for your sample, which is 10 times smaller. Right. So, from the force curve, here, you know, if I have pushed it up and my signal is 1.2 volts, and 1.2 is corresponds to 1 volt, and 1 volt corresponds to 1 Newton, I hold it at 1 Newton. The same calibration curve will tell you, if I have to do this at 10 times less load, you will have another set point decided for it, and it will. So, once the cantilever is loaded, you can always change the PID reference here. This reference can be either put 1 volt or 0.1 volt. If I put 1 volt, 1 Newton, 0.1, 100 piconewtons. So, if it is 100 piconewtons, along that curve you will either be on this side or that side, more load or less load. So, when approached and locked into feedback, I can change the reference value to push and pull, okay, and at whatever value you want to take image is your choice. No, this is just contact mechanics.
But still there will be charges over the molecule.
Yeah, if there are charges, then it becomes complicated, where there is a electrostatic interaction. You would like to use something called electrostatic force microscopy, or KPM, or Kelvin probe, or a potential difference, but that will involve biasing the tip with respect to sample, and if there are local charges and whatever, all that is different. Yeah. Yes. So, suppose that's one special thing, like membrane potential gap. There is a very special AFM that you have to use. What we are talking about is very simple cell with the topography is mainly the driving the whole thing. The contact mechanics between tip and sample is driving the interaction, but many times that is not the case. It is only could be this electrostatic single molecule interaction, and more will happen as we progress. But let us understand the basic topography imaging using AFM. Yes, but what is dominant is the question. Suppose I load it by Young's model, contact mechanics to a large extent, all these interactions will be minimal. So, we have to choose also. Any other question on topography? Contact mode. This is called contact mode topography imaging. Do it in liquid, do it in air, does not make any difference, everything is same. Any doubt? Yeah.
So, I mean, as you said that the height difference is corrected over here. So,
Yeah.
what is the range till which we can, I mean,
That
how
depends on how well tuned your PID is. So in a PID, there is a proportion integration derivative. So proportion gain, if you keep it too large and your topography also is too much up and down, then whole thing will be a problem. So if the topography is too large, you keep the proportion gain low, so that will accommodate that height difference. So that is all the game of adjusting the PID. Yeah.
environmental factors like when you choose the sample analysis, if this is not done in the vacuum, right? So there will be some environmental molecules that will be interacting with the thing.
Now, so those interactions are quite tiny. So if I load the material with about 1 nm force, and I'll tell you the interaction between proteins and this and that is in the range of 1 to 10 piconewtons, going up to 100. That is the largest, and 1 nm is the largest load on the lever. Okay, so for imaging, we typically so this one works. Yeah, good. So that's the bottom line. Yeah, one more question. Yeah.
Now, in case of any of this, whatsoever is my electronics and all, I'm going to get a signal,
Yeah.
whatsoever the topography, because the topography is not revealed. So how do I go for, understanding that what is a noise and what actually is my
Is
signal?
from PID scanning, particularly
Yes,
PID may respond slower,
yes.
faster. While going on the edge, the edge is sharp, so it's quickly changing from 1 volt to 1.2, as we said. So the sharp edge will not reveal itself as sharp. So how do you tackle this? So the point is, if you go scan this way and you come back also, right, on the next line, and between two lines there is hardly any topography difference, and they should fall on each other. That's when your PID is tuned perfectly.
But do you use some standards also before doing, for
Yes, of
example?
course, there
There
are
are
these,
standard
yes,
samples
yes, there are
also, they
these
are being used.
gratings which are
So,
nanoscale gratings.
so they
You
are first
image,
compared
yes, yes,
before testing the
yes,
sample.
you image them and tune your PID controller so that, as I said, while going this way, the topography line should match while coming this way. And of course, what will happen while going this way, this topography will look little different, and while coming, that is what will appear. So that difference will happen little bit, but mostly should match to each other.
One
Then,
more question is that this, related to noise and all. But the thing is that how it is ensured that there is no electronic noise in it,
these are
besides.
the standard the games of the instrumentation. You use the same thumb rules of instrumentation. Maybe you can spend little more
What are
time
the
in my.
typical SN signal to noise ratios involved?
It depends. I mean, what kind of cantilevers you use. You have to make
What
sure
kind
that
of
noise
sample is
is,
being
yeah.
scanned
The
also?
point is this noise that I just talked about, which is relating half k mean square displacement to kT. If that is the only noise source, that is the inherent thing that coming out of cantilever stiffness. You can't go beyond that unless you use some squeeze states or whatever quantum mechanics to cool it down. But to that limit, if you are getting thermally limited measurement, you should be happy in AFM. So that's what it
Thank you.
is. Yeah.
Sir, so you had just said that mapping the topography of the sample that can be done in liquid or in air. But if we are trying to map the topography in a liquid medium, then will not the fluidity of the liquid itself, like
Fluidity, okay.
will be sensed more by the tip with respect to the topography of the actual sample?
This is a concern when you do high speed AFM, as we were just talking about, and this is the ways to mitigate these problems. But when you are scanning at this rate, it's not such a big problem. It's all you have to see, the fluid really becomes turbulent and all, it's a Reynolds number, no?
No, I'm just talking of a simple buffer solution.
Yeah, simple buffer, as long as your movement remains within laminar, as we call in physics, which doesn't cause any turbulence, you are okay. But your movements is rapid scanning is like stirring a water, that kind of a thing if you are doing, then problem.
Okay.
As long as you are doing this slowly, air and liquid and whatever doesn't make any difference to the cantilever.
And like that probe will
Yeah.
still not will not come in like contact with the liquid, or
The
it
whole
might
thing is
get-
immersed in liquid when you do liquid imaging.
including
Whole thing,
including the tip-
cantilever, everything is immersed in liquid. When you do liquid imaging, the whole thing is in liquid.
Okay.
Okay, so any other questions on topography? Yeah. Yes, tip modifications is a large issue. That's what I showed you in the tip that is used once looks dirty. Soft sample is not a problem. Stiff samples, tip can get damaged. But again, it's also load. How much load you apply? If you apply too much load, then it may have a wear while scanning.
Like when the starting point, it will be very nifty, but while it is ending the same.
Yeah, so throughout your imaging, if you use soft samples, tip will not get damaged per se, get dirty. If you use hard samples, then tip may get damaged even while scanning, may get modified. It becomes little flatter. Yeah.
Liquid of any viscosity or density can be used for.
No, buffer is okay. Glycerol and all very high density things one should not use. Yeah. Within some range is okay, but not too dense like honey, no. People have tried these things in tribology, which is this friction measurement business, where oils are different viscosity are used for friction, but then they are specialized applications, I would say. Normally, if you have AFM want to do biology, do in buffer. Don't try to change viscosity by many orders. Okay, so that was about imaging. Second thing I said, force spectroscopy, the very thing that we used here first to decide what we load the thing with. That is our what is our force curve. Use that for deciding what force we should apply while we image. But this force curve itself, suppose I took an image and I got some topography, and I now say, yeah, fine, I got this topography, but I am clearly interested in how soft or hard this book is compared to there. Okay. And this then can be done by, suppose there is some feature on this image which I want to probe into, poke compared to there. So, I will take one force curve there, one force curve there. This is used for calibration, which is basically, we will explain that in a demo session how the calibration works. And now, when I press on this, it will, let us say, and this is a stiff enough lever that does not cause any jump in or attractive force, does not sense attractive force, it goes like that. Okay. And if it is here, it will go like that, but suppose this is soft sample, on that it will start 0 here, again touch at the same point, but it will slowly go like that. This is hard sample with a higher Young's modulus. This is a soft one like a cell compared to glass. And now if I do a pressing there and pressing here, based on this difference in a way, I can say what is the Young's modulus here. Okay. And this is what is used for cell mechanics, we will talk about it more later. So, that is about contact. So, you can use contact mechanics to determine nanomechanical properties. You can make a whole map, I can take one force curve there, one here, one throughout the whole thing, and say, okay, this cell is like this, a nucleus inside which is also different stiffness. Locally the stiffness can vary, I press there all over the place. I can get a nanomechanical map of this whole thing. Okay, so that's one thing that you can do with the force spectroscopy measurement, measuring the mechanics of while you are pushing on the surface. And second, very important for biomedical applications and other things is single molecular interactions. Okay. Now, this is the part which we will talk about more in depth, but suppose I have a, suppose once again I take this example of this chair, and I suppose I have this these are the protein molecules which are sparsely decorated on the surface, which means if I do this, then I not really able to see one from the other, right, difference. But suppose I sparsely put them up, like that, and I come down with a tip which looks like a sharp thing, but the tip radius is 10 nanometers, I say. I mean, this could be 1 nanometer protein or 4 nanometer, and this means that the tip is like huge object, and which comes and presses on the single protein, and then you attach it there by tip functionalization strategies, we will talk about them, and then pull on this, and this is like protein that can unfold. Okay. And I can have a sequence of proteins that unfold sequentially. So, what is the idea? You go in like that, which will appear like a normal curve, which will show nothing. At this point you are pressing on a protein, but it is such a small thing compared to the tip and the substrate and the whole thing, nothing much will come out of it. So, you press and you sort of go there at maybe 100 piconewton, 500 piconewton, and stop there for a while, so that your tip makes good interaction with the surface of the protein, gets linked to it, covalently attached or whatever. That reaction takes time. So, you wait for maybe 5 seconds or 2 seconds, or 1, whatever, depending on the rate constants. You do that, and now it is attached. One end of the protein is attached to the surface similarly already when it is falling. The other end of the protein, N or C terminus as it is called, is now linked to the tip, and you start pulling. So, two sides covalently attached, and you pull on a protein, it will unfold. Okay. So, what will happen? So, this goes up, and now while coming back there is a protein, and which will resist its unfolding. So, this will appear as a loading with a force. It will have its own characteristic, and at one point it will unfold, and this will, once the protein unfolds, it has lot of length, it is 4 nanometer protein can become 30 nanometer protein or 40 nanometer, and then because it is resisted pulling, cantilever is pulled down with it, and at one moment unfolds, the cantilever goes back to 0, and then you can keep pulling this further, this guy further, and finally it will detach from one end or the other. So, this kind of a curve you can obtain. Suppose I put sequence of proteins, maybe with linked whatever, okay, and this is the tip. I came all the way down, and this is now I am showing as an unfolded thing, but this will be lying on a surface, right, and then I picked it up and I pull this down, and this will, one will unfold, and second will unfold, and third will unfold. As you keep pulling, each time when one domain unfolds, the cantilever that has bent back will go back to 0, and then next one will, you start loading, it will come down and go back to 0, and so this will provide you a profile that looks like this. Okay, and if you have 4, there will be 4 such. If you have 8, there will be 8 such domains. What does each one signify? Drop in force from negative value to 0. That drop in force will mean that one event, unfolding event has happened in a protein. And after going up to here, we still have, let us say, 5 domains unfolded and total 6. You can still do a refolding experiment. Okay. You pulled and 6 out of 6 domains, 5 are unfolded. Now I say stop, just not pull any further. We will go back and see, as you go back, they will refold into the between the, there is no force now on it. And this refolding also can be, so folding, refolding, unfolding and refolding can be done with AFM, and that is the force distance mapping. Yeah. Yeah, just give me 1 minute. Now that's the unfolding, refolding thing. What can also be done is that I have this receptor or protein that is of interest, and I have uh, Peter will talk about it more. The most famous example I can think of is corona. On a cell surface, you have S2 protein. That's the receptor of binding domain of the RBD, receptor binding domain of the corona, which is a spike. That's why it looks like corona. That guy attaches to the receptor on the cell. Now you can forget the cell, you can forget the virus, just get these two, isolate them, put one on the tip. So attach chemically you. So now you have attached some with a linker, kind of a peg linker, and let's say I am showing this as they normally show, no, as a thing. Let's say this is a kind of thing that will be there, and you sort of have on the protein something that will fit into it. Yeah. So what we have is we have linker with this receptor binding domain. Receptor will be sitting on a surface or on a cell surface. You bring the cantilever, let it interact, and then you start pulling. What will happen? The cantilever will bend. It will measure the force required then to unbind them from each other, and this kind of thing will appear as you go in, you come, and there are these linker molecules have, let's say this is a 10 nanometer linker, and similarly this also is 10 nanometer linker. So total thing that it really starts to apply a serious force on this one is 20 nanometers. So you will start seeing this kind of force curve, where this is the rupture of these two, separating them from each other, and this is the linker's elasticity. You know how linker will extend under force, and that's what it gives here. And this is then is called a receptor ligand dissociation. You do this at different loading rates, do pull this at different speeds, and how do you get the binding constant of one protein with another? Is on rate, off rate chemically, what is the barrier height for this to happen, all of this we will talk later, but this is how force spectroscopy is done. Any doubt on force spectroscopy? You had some question. Yeah. 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 Yeah.
The thing is that we don't know whether the protein molecule is like this or like this.
Right.
So
It's
how would you ensure that it is
Right. Right. Yeah,
connecting, the tip is connecting, that
yeah,
is thing.
that's a good question.
Then isn't it a very tedious process also?
Yes.
Parallel, because
Extremely tedious.
being able to identify exactly where the molecules are, and to be able to pick them, and to be able to
It's a fishing, I will let's say. So I'll first answer the first part of the question was orientation. It's very important question in doing all of this. So for instance, if I am pulling on this protein this way or this way, or this protein is lying like that and I come down and I picked it up and it then came like that, or I actually held up on something, some chemical group here, and now I am, instead of pulling on this that way, I am pulling laterally. So if you are basically, I am pulling on this side or pulling vertically or laterally, all of this depend on the orientation in which the molecule is lying on the surface, right. So there are a couple of things that are done for this particular question. What you do, you take a protein and put something called his tag or whatever is normally done, and that is like a 6 his groups or cysteine groups. If you are doing gold tip and this thing, put extra cysteine groups at the end, and so you got N terminus and C terminus. You add amino acids which will be happy with substrate, one end substrate, the other end will be happy with the tip. So now when you come down and this guy is lying like that and I am trying to search, it will reorient itself or whatever to attach to that particular N or C end of the of the protein. So, you are basically doing end to end pulling always, but that does not solve the problem, because if you really think about proteins unfolding, it may unfold under a force which is much lower compared to you do end to end. Okay, because we thinking about end to end, but when the process happens in biology, maybe like when degradation of protein happens, it has to be unfolded. It happens at a much lower force, because then it knows exactly pulling along which two directions will unfold it easily than pulling along end to end. So, some work has been done on a lot of work has been done on putting certain groups on the protein, so that your pulling becomes in different directions, and yes, it is anisotropic. The protein pulled in one direction as opposed to another shows completely different stiffness. It unfolds at much lower or much higher depending on which direction you pull. That was a question number 1. Second is, it is extremely tedious. Of course, it is very tedious. The data analysis also later on is tedious. What we are doing is basically like you guys are sitting in a room, you are too many actually, because the tip size is for this room is more than the room size. So, I got protein with this concentration, it is no use because my tip will pull every one of you. Okay. So, it is difficult to say this is not a single molecule experiment, then I am not isolating. So, what is the idea? Probably one guy should be sitting in this room, another room, and each time I come down. So, you got a sparse decoration of the protein molecules, and how sparse it should be, that my tip comes and picks this guy up, this guy should be far away, away enough, so that when I do this guy is not disturbed. Okay. So, as a result, you have to use very low concentration when you put the protein on the surface, and then you do a fishing experiment. So, what do? You say, okay, I will select this grid, okay, 60 by 64 by 64, 8 by 8, whatever, do one experiment there, and the next one is done maybe 5 nanometers away, because that is the tip more than tip contact area, and then third one is another 5 nanometer away, and this way you scan the whole thing, you will maybe have a protein there, here, there. Okay. Not everywhere, it is just to partially put on the surface, and as a result you take 1000 force curves and maybe how many? 4% or so. So, if you want really good statistics of all this data, reproducibility, everything, after 1000 force curves you get about good enough statistics, which means you get 40-50 curves, or if you are lucky you have a 10% success rate. So, it is a success rate. So, it is extremely tedious. Once you take those good, and how do you identify good curves from bad? Suppose there are 5 domains in a protein that I am expecting to unfold, you just look at only those force curves which are showing 5 events. That means I am really pulling on what I intend to pull on, and throw away the rest. Only 2 domains you are seeing, do not throw away, because you are not doing end to end pulling.
What about the negative? For example, rest of the say 96% of the experiments they are so they are from the experiment viewpoint, they are negative results.
They are basically, it is what I am saying is, suppose this is a fishing thing. You sort of doing fishing for long and nothing comes there.
But that data also is something.
It is only giving you mechanics of the
surface.
Yeah, and it is not telling you anything more. So, only mechanics of the surface is what you are getting, because there is no negative force, there is nothing in the downwards pulling.
No, you are looking for an event.
Yeah.
So, those events only you
consider. Those are only considered as events, and statistics.
Folding, unfolding parts.
Yeah. Others is just, it is not negative that I would say, it just something.
For example, a protein being selected.
Yeah.
But not exactly as per the requirement of the experiment, and that does not bring in all what is. But
so, for instance, like you have got a fragments of protein.
Correct.
You got fragments of other proteins which are not, while in a process of purification, you get something also, a lot of dirt, and you pull on them, and it shows you unfolding events, but they are not 8. Like typically you engineer a 8 domain protein, and you have to see 8 identical domains, because you engineer 8 identical domains into protein. Those only are considered. Other thing is considered as junk. It could be anything. So one thing that people use also, 4 and above is considered, and sometimes 8 and above is considered, but depends on you. Yeah. So that's a way to go about it. Yeah. Yeah. The
So there are multiple interactions between the molecule or among the molecules,
Right.
which we are trying to
Right, yeah.
see.
right. Like
So
ligand receptor is between molecules,
yes,
like
yes. So
receptor and
when you're
bind.
applying force,
Yeah.
pulling force, so one would speculate that the breakage of interactions would be gradual.
Uh-huh.
So do you see always that kind of curve, like a snap?
Right. So suppose you got a multi-domain protein,
Mhm.
and now here let's say I engineer a protein with 8 domains, identical domains, and I see all identical things. But something like bacterodopsin, which a lot of experiment people have done. So bacterodopsin sitting in a membrane, and then you pull on it, and it's got 5-6 domains, and some of them unfold together, some of them unfold one after later, and some third. So you typically see 4 unfolding events in bacterodopsin, because 2 are so tightly together they unfold, and then the rest one unfolds, and then third one, like that. So you get a pattern of bacterodopsin, which is also done with a lot of experiments, and people have what they have done is put a bacterodopsin there, take an image, and then pull a bacterodopsin, it comes out of that membrane, and then you take an image again, that rhodopsin is not there, which means the previous time you pulled was actually bacterodopsin. That kind of thing people have done. So this is in between domains, the interaction is probed this way in a sequential unfolding. About hydrogen bonds, there if you look at the amount of energy, bond energy that we have is much less than our sensitivity limits. All AFM optical tweezers or magnetic tweezers, it is much below that. So individual hydrogen bond force required to rupture them is not yet measured, and it happens in such a stochastic manner. Suppose I got 2 beta sheets connected by hydrogen bonds, and I unwrap them, which we do in the lab. Then is it possible to say, okay, now that one gone, this one gone, and that one gone, and will that happen always in the same sequence? It's difficult to say. So it's very difficult to, in for spectroscopy business like in NMR or any other, it is very difficult to assign rupture of hydrogen bonds in the experiment. The signatures won't come.
So most of the time you get one.
Yeah. So for instance, let's say I have a protein that is, let's say, titin we do in the lab. So titin has, and everyone knows this, and we have also measured, is beta sheets. Okay, and at 100 piconewton force, one of the beta sheets falls out by breaking 4 hydrogen bonds. Okay, so it comes out, and then what happens is the remaining part is again a protein. So what you get is that signature of, yeah, first that has come out and later. So in a force curve, you will see something like this. So this is one, and that's another. So this is where and you can also see the length of this equal to exactly the amount of number of amino acids that have unraveled by unfolding one beta sheet alone. Maybe 6 amino acids gone, and 6 amino acids put next to each other will contribute to that difference. So it is captured that way, but individual hydrogen bonds difficult. So this we call as a mechanical intermediate, which never can be captured in biochemical reaction. This is one strong point. Particularly proteins under mechanical force better be studied with mechanics, and doing a biochemical reaction such as not normal protein folding unfolding is actually doesn't give you much, all this kind of information.
Thank
Yeah.
you.
Sanket.
So two questions. How do we know if there is single molecule attached on the tip? Is there any way to find it out? And the other thing is, if we repeat this folding unfolding experiments by approaching the cantilever back and
Refolding,
forth.
yeah.
Yeah, yeah.
Folding, refolding, yeah.
So,
Unfolding, refolding.
like generally in condensed matter measurements, we take multiple force curves and take some averaging or
Right.
to remove noise.
Yeah, yeah.
But I guess will heterogeneity play a role and do the
Right,
folding
that is
happens?
always a problem, because this happens in condensed matter experiments. You have a Avogadro number of molecules, so the degrees of freedom are huge. It's a continuum thing. Here it's a single molecule mechanics, so which means it will not be same every time you do it. So it is there is a large amount of, the stochasticity plays a role, and the branch of stochastic thermodynamics is more appropriate. So what you normally do to do this, to address this, is load this at a different rates, unfold at different rates every time you, and from there you can apply some of this Bailey-Vance theory we will talk about later on, to extract out, tease out. Because see, when in corona for instance, receptor binds to the binding RBD binds to a receptor, it also at no force it will bind, unbind, bind, unbind, keep doing that. The point is to figure out how long it stays in a bind state compared to unbind, and one unbinding experiment does not tell you what is the force required to unbind them. So you do this at a different loading rates, and then apply Bailey-Vance model to tease out what was the dwell time from stochastic thermodynamics consideration, and that actually works pretty well. Peter will show you very nice examples from different variants of corona, Wuhan, Micron, Delta, and how the what do you say, the spread of disease, what is called the infectivity, is more for one compared to another based on these measurements. I mean, you can see this correlation. Also, their high speed image, and that will come later. Yeah, but it's a single molecule, so don't have the luxury of typical condensed matter experiments. Yeah.
What about the how do we know if there is single molecule on the tip attached?
So this, for instance, as I said, if I do this, two parallel, one will rupture, then the second will rupture, that will have a signature of that kind. Sometimes you take a force curve and you see 8 unfolding events, and together two domains have simultaneously unfolded, so you get a contour length which is twice as much, but one large peak instead of two. And many times if you have more domains, then the force curve will not look typically the way it should look, and then one can say, yeah, this we should not consider, and that happens when you are playing with too much concentration and stuff. So you look at that part is quite tedious to sift data from garbage in a way. Yeah. Huh.
What are the typical lengths of the time lengths?
Okay, so the thing is, you can, for biological samples like protein and things, you put them on surface, and in buffer conditions and whatever, you cannot exceed the experiment time more than 3 or to 4 hours. So in a day, everything is working well, and experiment starts to happen, you get 3-hour session to do this, and in that you can maybe grab 1000 curves or whatever to generate a statistic. So on a lucky day, you will get good enough statistics for that pooling rate. Next day you will do another pooling rate, and so you have to do multiple pooling rates, pooling speeds. So to address this question which just was raised by Sanket, that how do we take, so you need that data for over long, maybe 2 orders of magnitude pooling rates to make sense of the whole data. So to finish and make conclusion after analysis may take couple of months of good days of data collection. Together that will amount to 6 months maybe. So it's a tedious and long procedure, but if you do a streamlined way of functioning, it does give you good data.
Second question was, for example, before deciding where are the possible, protein samples available, which could be linked. So how long it takes just to scan?
One image? The normal AFM image takes minutes.
Huh?
But then Peter will talk about high speed AFM, which happens at the dynamics at the molecule scale, even milliseconds.
Okay, that is
So
what
this
I
is
was.
one branch of AFM which is called high speed AFM. And last week me and Peter were in this meeting in Madrid. Toshio Ando is the founder of the high speed AFM. They have pushed it to the limit now. So you can actually get molecular interactions happening under AFM being
Oh,
seen.
great.
So that the speed, and those AFMs are only specialized high speed AFM for imaging molecular level interactions, proteins dancing on the surface, antibodies doing whatever, all that. Yeah.
So this second question was related to, can some other technique like interferometric microscopy be coupled with this?
Oh yeah, this is called correlative microscope.
Coupling.
Yes, this is called correlative microscopes. A lot is being done. So you
Thank you.
attach AFM with FCS we did in our lab, AFM with confocal, AFM with STAID microscopes, AFM with, so these are correlative microscopes. Then AFM with SEM and TM. That's happening with piezoresistive thing, and group in I guess EPFL is doing this. So a lot of things, yeah, a lot of integration is always on, yeah, that gives you more. People do that, yeah.
Thank you.
Just will come up.
From data to protein structure, it seems like a inverse problem.
No, we don't get structure.
The domains basically.
Yeah, this we measure force required to unfold a protein or force required to rupture a bond,
Okay.
and done at a different loading rates. This can provide you crucial information about unfolding rates or rupture rates.
Okay.
Okay, so that will give you something about how strong the bond is or how deep in the energy landscape protein's folded structure is,
Okay.
like that. But it does not tell you, like a cryo EM or something, it does not tell you, it doesn't resolve structure of a protein.
I see. I see.
Yeah. And it's not, because it's not imaging, it is just mechanically applying force and seeing how much force required to do this and do that. So if you want structure, then cryo EM, not AFM.
No, because I was thinking in the direction that since we are facing a lot of problem because the domains we attach to, and since we have a
Maybe if it's a research related specific
Yeah.
problem in a lab, we will talk about it in lunch, during lunch or even after. Yeah.
So I just wanted to know this for contour scanning and force measurement, is the cantilever different?
No, same cantilever.
Same
Same. So only difference is that probably for imaging, contact mode imaging is always done in liquid. Contact mode imaging always have a cantilevers with 0.1 Newton per meter is stiff end all the way to 0.001. So these kind of cantilevers are used for contact mode imaging in liquid because you don't want to spoil the sample cells such as not much load, and for unfolding also similar cantilevers are used. So that's similar. For ambient conditions and something called ultra vacuum imaging, we use cantilevers which are 100 newtons per meter, 50 to 100 newtons per meter. So that is different. But in liquid, doing similar kind of samples, soft biological samples, polymers, that's the cantilever typically used. Yeah.
So, like, suppose I have two single domain proteins, one is A and one is B, suppose. So, I do the mechanical unfolding by AFM of both the proteins and I get the force extension curves.
Yeah.
So, based on what specific factors do I look in the force extension curve that will give me some information about that which protein is more stable? Like, what should I be actually looking
Mechanically
into?
more stable. Which one of the two?
Yes, sir. Means like saying that both are single domain proteins.
Sure. So if you get two unfolding events, and one will require more force to unfold than the other one. Now, which one unfolds first and which one second is something you have to get other information, maybe from structural or other things. So, the B has, let us say, some clue about the B is more stable mechanically than A, then B will always have more unfolding force than the A, but how much difference is what AFM can tell you. But A is stiffer than B or B is stiffer than A, that at all is not known to you. You just pull, one guy will unfold. Will this unfold or that unfold? You don't know. So, you have some ideas to, okay, this guy is necessarily little stiffer than that, but how much stiffer or how much more stable mechanically, that relative information AFM can provide.
So like that prior idea that which protein is more stable, that
Yeah.
I can get from the melt curves of both the
Yeah, that those many structural information,
be
alpha fold nowadays, you put the sequence, can give you some idea about
that
which one.
gives a prediction.
Some prediction, that's what I am saying. So, some idea if you have, then you can proceed. It's, yeah.
What is the ideal dilution you required for that, or optimization again we have to do?
So, depends on molecular weight, but our typical thing is 1 microgram per microliter. Where is so now? 1 microgram per microliter, but that microgram or 1 or 2 is depends on the molecular weight, but extremely dilute. You don't need large amounts of protein to do AFM experiments. Even 10 microgram protein can give 10 experiments, AFM experiments. Yeah.
Hello. So
1 microgram per ml, that's control. 1 microgram per ml, roughly. Yeah. So question.
so these kind of force curves which we get for linear elastic materials, like having a single
Yes.
elasticity.
Yeah.
Oh, how to do like force spectroscopy for gradient stuff?
Materials with non-linear.
Like
Well,
if-
single polymer chain is a non-linear.
Yeah, example, I'm making a hydrogel having a gradient elastic curve.
Right.
So how to do force spectroscopy of that, and what
If
kind
you
of
take a hydrogel and you want to press on it and see the in contact part how this works,
yeah.
we'll talk about it in my lecture number, whatever. And we do this in lab quite regularly.
Sure.
We do it on cell, but also we do on gels.
Okay, like in generally cells are not linearly elastic. They
Sure,
are
all
more
of that. Constitutive
viscoelastic.
equation of, you have to take a constitutive equation assuming a standard linear solid or a power law rheology or a Kelvin-Voigt or whatever, or in contact mechanics you do DMT, JKR, all that.
Yeah, like
You
linear.
can pick up a constitutive equation to describe the interaction between tip and sample, and then on top of that the protocol of measurement to reveal the G prime, G double prime, or for our whatever the power law exponents. Then we do that.
Yeah, please brief on
yeah,
that.
yeah. So you will see.
Thank you.
So there are no questions. I wanted to speak more about the dynamic mode AFM. What is the time now? I think it's lunch time already. Am I right? What time is it? 1:10. So I think we should stop here. The two more things that I wanted to speak about and I didn't get time is the dynamic AFM. When we oscillate the cantilever and tapping mode, you must have heard about and all that. I will talk about it maybe during tutorial session today a little bit, and we will then discuss more. We'll maybe take 1 or 2 examples of cantilever calibration, how calibration is done, and the other group, 2 batches, no, A and B. Other group does the lab session, and you come back after snack break, and like that. Okay. So let's go for lunch now and we can talk more there.